Aloha and welcome to our video on the structure of DNA. The goals for the video are to describe the structure of nucleotides, and then also we want to describe the structure of a DNA molecule. Okay, so let's start looking at the parts of DNA. Now, DNA is a polymer, and if you recall back, we were talking about polymers several times. We said a polymer is a string of molecules, and these little molecules that they're a string of are our monomers, and in the case of DNA, it is a nucleotide. So here we have a picture of what our nucleotide is. If you notice, there's a phosphate group here. We have a sugar, and the sugar is going to help us determine which type of nucleic acid that we have, whether it's DNA or RNA. And then we have our nitrogenous base. And our nitrogenous base is what's going to code for the proteins, and we'll learn about that a little bit later on in the unit. So when we take our nucleotides and we join them together, you'll notice that we have one of them is here, and then another one is down here, and then another one here, and you can see how it's this string together effect of a polymer. And they're joined here with the sugars and the phosphates. The sugar of one nucleotide will join with the phosphate of another nucleotide, and this is our bonding here that makes this polymer grow. So as I hinted, we have two different types of nucleic acids. We have our DNA and our RNA. Our DNA, they both have the phosphate groups. Our DNA gets its D from deoxyribose, and our RNA gets its R from this ribose sugar here. So our sugar determines what it is. And then when we look at our nitrogenous base pairs, they're all going to contain adenine, cytosine, and guanine, but our DNA will have thymine, and our RNA switches it out into uracil. So that's just a real quick overview of the parts of DNA. Remember, DNA is the polymer. Okay, RNA is also going to be a polymer, and both of those are going to be made up of these units here we call nucleotides. Okay, let's meet the nucleotides here. Notice that we have two different fundamental structures. We have our two-ring nucleotides up on the top here, and then we have our single ring down here. Now, if you notice, there are 5 nucleotides. We are going to have some that are going to be in DNA. We'll have some that are going to be in an RNA, and the bulk of them, 3 of them, are actually going to be in both. So let's take a look at DNA first. DNA is going to have the guanine and the cytosine, as does RNA. RNA is going to have guanine and cytosine. Both DNA and RNA are going to have adenine. But here's where we see the difference. With DNA, we use this base pair thymine, but when we get to RNA, that thymine is switched out to uracil. And that's one way we can tell if we have DNA or RNA is by looking at the bases. And if we have the uracil, it tells us we have RNA. And if we have this thymine, then we know we have DNA. Now, the discovery of DNA structure is kind of an interesting tale. It talks about 5 different scientists that we want to mention real quickly. One of the first was Erwin Chargaff, and we'll talk about him real quickly. You can see a picture of him over here. What Erwin Chargaff realized was that when he took DNA and he broke it down and measured its components, he realized that the amount of adenine was always equal to the amount of thymine. He also found that the amount of guanine is also equal to the amount of cytosine. And when we look at the structure, we'll kind of see how that starts to work out. Now, we also get to meet these 2 individuals here, Wilkins and Franklin. Franklin probably is the least known of all of these, but lately she's gotten the press that she's been due. What these 2 individuals did is they took a picture, an X-ray picture of DNA, and saw that its structure. And by looking at that picture, they inferred that it had to be some kind of a spherical or helical structure. So they had that information. At the same time, Watson and Crick, who got most of the fame for determining the structure of DNA, they were building models and trying to make this work out. And what they noticed is when they saw this picture here, they realized what it was, that it would be a double helix. So there were 2 strands in DNA. So DNA has 2 strands, and those 2 strands are going to be twisted on each other. Okay, and then what they also started to look at is they started to look at, from the x-rays, how wide these 2 strands had to be, and they came to the realization that if they took a purine, which is a double ring nucleotide, okay, so you can see 1 ring and 2 ring over here, and a pyrimidine, which is a single ring, if they took 2 of these purines together, it was too wide, If they had two of these single rings, it was too narrow, and the only way it made it fit the data is if they had one of each. So if we go back and we look, we notice that we had our two ring structures and our one ring structure, and those ones here, each of them had a complementary base pair, and we can start to see from Chargraph's work what these base pairs are going to start being here. So finally, we got to what we know DNA looks like. We have our complementary base pairs in DNA where we have adenine and thymine, and we have guanine and cytosine. So when we notice and we have our two strands here, you see our strand 1 and strand 2 there, what we notice is these ones will always bond to each other. So we always have the adenine thymine bond, and we always have the guanine cytosine bond. Now, if you recall back to our earlier slides, we saw this sugar phosphate backbone, and that builds up this edge, this rail, so to speak. And we'll notice that in both strands, and those strands are going to twist behind. And a lot of times it's like a spiral staircase. So the rails of the staircase are going to be this sugar phosphate. And then we'll notice that the steps themselves are going to be these actual bonds of the base pairs. And that's what our DNA structure looks like. You can kind of see it over here as well. It's a double helix. And the lessons will go into it a little bit more for you. Good luck on the lessons. And as always, we'll see you in the next video.